Hello again. Welcome. My name is Katie Barron Nalvin. I'm one of the board members of the Cape Perpetua Collaborative, and I'm really proud and excited to be here today to introduce today's young wave maker, Charles Nye. But before I do, I'd love to just take a moment, give you some quick background about Cape Perpetua Collaborative for those who might not be familiar with us. Um, so we're a small community organization operating in the Cape Perpetua region. So that's roughly speaking about like the shoreline and the adjacent forests to Oregon's largest marine reserve, the Cape Perpetua Marine Reserve. Our mission at the Cape Perpetua Collaborative is to cultivate collaboration across communities, disciplines, and generations to inspire care, understanding, and action in alignment with the ecosystems and cultures of Cape Perpetua from the land to the sea. So once again, super thankful you're all here. Our Young Wavemaker series is designed to be a platform for young or early career individuals to showcase their conservation work relating to the Cape Perpetua region or those that live in the area. So this program is entirely supported by small dollar donations. To support our work, please consider visiting our website and click the donate button, or show your support in other ways. Contact us for other ways you can get involved. Also, just a fun plug there too. We sell great merchandise. Highly recommend it. Anyways, swim on over to our website. It's all there. So in today's webinar, Charles here will give a presentation followed by some discussion. So if you have any questions, please just type them into the chat or use the Q&A function. I'll be monitoring both. And at the end too, if there's time, I have some questions to ask Charles as well during our discussion. So to introduce our young wave maker, Charles Nye, he is a wildlife science PhD candidate at Oregon State University. He is a marine conservation geneticist, which means he uses DNA to better understand the diversity and ecology of marine life. Charles applies these methods to research cetaceans, whales, and dolphins, along with their prey and other species they share their environment with. Charles received his bachelor's in marine science from California State University in Monterey Bay in 2018. Before starting his PhD, Charles worked at the Monterey Bay Aquarium and the Monterey Bay Aquarium Research Institute. In addition to academics, Charles is an avid scientific illustrator, tide pooler, and coffee connoisseur. So Charles, off to you. Feel free to share your screen and get going when you're ready.
All right, let me go ahead and do the thing and ask the question that everyone has heard since COVID. Can you see my screen? We good?
We're good.
All right. Well, good afternoon, everyone. Thank you so much for joining us today. As Katie mentioned, my name is Charles Nye, and we're going to be looking into this thing called environmental DNA and how that helps us look at whales, solve mysteries about whales, and how this generates more questions about these species. So we already went through who I am and what I've done, but I would be remiss not to introduce my lab. So as a PhD candidate, I am part of a research group, Dissertation Conservation and Genomics Lab at OSU. If you look at the photo in the far right corner over here, starting with this woman on the left-hand side, that's Angie Stremba. She's a research associate with us. There's me. Next to me is Keith Hernandez, one of our former postdocs. Scott Baker, our fearless leader and my advisor and boss. Kelly Lejovsky, a former master's student who worked on sperm whales. And last but not least, this is Debbie Steele, who I affectionately refer to as my laboratory auntie. She's our research technician and kind of the heart and soul of what we do. So my research interests are very broadly about what lives in the ocean. I'm very interested in biodiversity. How much of things live where? When do they pop up? When do they leave? How many are there? And this also ties in closely to evolutionary biology. I'm just fascinated with the diversity of life on Earth, past and present, and kind of trying to disentangle those relationships as well as understanding what it means to lose diversity, right? As we're facing an extinction crisis right now on Earth. And some other career things about me. I'm a scientific illustrator. I specialize in paleo art. I also draw whales and dolphins. I like drawing big things. It's just something about the physiology of huge creatures that fascinates me. This pushes life to its limits. And also, I'm an avid proponent of scientific communication, and this is sort of why I'm here today, right? I'm just a guy. Scientists, very few of us are like lab coats in ivory towers in closed door rooms. Most of us are just people. And I just want to tell you what I work on because I love it, and I want to share why I love it and why it's important. Much like other people in my discipline, this goes way back, right? I think I have some family members in the audience, but they will be able to corroborate that this goes deep. I've always loved whales, dolphins, the ocean, elephants, dinosaurs, you name it, right? I've had a fascination with nature for as long as I could imagine, but I didn't know exactly what that meant for a career, you know? When I started my undergrad years, I didn't quite understand what a marine biologist did, right? And so I did kind of everything. I got scuba certified. I did like zooplankton monitoring and collections. I volunteered as a docent at the Monterey Bay Aquarium. And I also did like things like specimen prep and museum skin stuffing and stuff like that. It wasn't until my very last year at CSU Monterey Bay that I actually found the hook for my career. CSU Monterey Bay has a research program for undergraduate students. It's called UROC. I applied for this program, got accepted. They give me a stipend. They pair me up with a researcher, and off we go. I was paired with the great and late Dr. Eric Crandall in his molecular ecology lab. And we looked at invertebrate genetics, things like snails and sea stars and stuff like that, and how their genetic diversity changes over space, right? The further apart things are, the more divergent they will be are genetically, right? This is how speciation occurs. I thought genetics was too much for me initially. It's a lot, right? It's such a complicated process to learn about genetics, and not alone, let alone becoming an expert on something you can't even see, right? But to see how this was applied to conservation importance, right, kind of sparked that light bulb in my head. The following year, I got employed by the Monterey Bay Aquarium Research Institute, or MBARI, where I got introduced to this idea called environmental DNA. I worked as a research technician, research associate in the biological oceanography group. We collected a bunch of seawater, did a bunch of experiments, but what is eDNA? What was I introduced to as part of this lab group? I want to do a bit of an interactive thing with everybody. I can't see you all, but I'm going to trust you're going to follow along. I want you to breathe in and now breathe out. You've just shed eDNA. Just by existing in the place that you live in, you are shedding eDNA. If you have a cat in the corner, it's shedding eDNA. If you have a fish in a tank, it's shedding eDNA in the tank, right? Organisms shed DNA in the places that they live. And we can collect that genetic information for all sorts of different kinds of studies. You could start to imagine some of the direct implications of collecting eDNA, biodiversity, public health, things like that. A single drop of water can contain the DNA of dozens, really thousands of species at a time. There's actually a lot of stakeholder interest in eDNA across the country and the world. I actually participated in the third national workshop on marine eDNA. This is a big meeting held out of the DC, Washington, DC area. It was hosted by Johns Hopkins APO, and then we did a last day at the Smithsonian. And when I went there, I got to interface with so many cool people, materials specialists, AUV engineers, geneticists, conservation workers, things like that, nonprofit organizations, universities. And I got to interface with so many representatives from different sectors and got to see how all this kind of fits together, right? And I regularly still talk with and interface with the people that I met during this meeting, which means that the networking prowess of this actually worked. You can't say that about a lot of conferences, so this one was a good one. And I am in this photo. If you look in the very far back, I'm in there with my advisor. There I am, and there's Scott Baker. I don't like being at the front of big group photos like this, but I'm there. How this works, though, for metabarcoding, I'm getting ahead of myself. How this works for eDNA, I'm using one facet called barcoding and metabarcoding. Consider the grocery store. I'm going to explain this. Please stay with me. You're at the store, you pick up your stuff. Each item has a barcode on it, right? It's a short sequence of numbers, and you scan it in. It's your tomato sauce, it's your beef chuck, it's your grapes, right? Everything on the planet has short diagnosable DNA sequences that are similar to barcodes in length. They're not giant strands of DNA. These are a couple hundred base pairs that are diagnosable across taxonomic groups. It's like I took a whale, found its barcode, and zipped it in, and I move on, right? And this is why we call them barcodes. Trying to get my screen caught up with me here. In practice, we call this DNA barcoding if it's one species at a time. We call this metabarcoding across many different species. And DNA, the kind of things that we use here, is really pattern recognition, right? There are four primary nucleobases, ACTG, adenine, cytosine, thymine, and guanine, right? And different combinations of those can produce species diagnostics. You're just trying to find patterns in this. DNA metabarcoding is what we use in eDNA. You have DNA sequences, DNA barcodes belonging to all these different species. You collect them all, and then you go through the process of annotating and finding all of those different sequences to get a biodiversity survey out of that. Now we're going to jump into the first kind of case study for today, but keep this in mind. We're using short DNA fragments to identify lots and lots of species in the ocean. The first case involves kind of the star of our ocean, the killer whale. In June 2022, I was working with the Marine Mammal Stranding Network here in Oregon. I still do work with them at a volunteer capacity, but they paid me to work with them for a summer. That summer, a dead killer whale was sighted off the coast of Newport, Oregon, which is where my campus, Hatfield Marine Science Center, is located. This is a very rare phenomenon. Killer whales do not entangle very easily, very often, and to my knowledge, this has happened very few times off the Oregon coast. The kind of gear it was trapped in is crabbing gear, the kind of long line attached to a heavy weighted net mesh kind of system. This research is actually published in Aquatic Mammals, so you can go ahead and have a read if you're a nerd like me and want to know about more of the specifics of this. Entanglements are a really big problem in cetacean and really just marine conservation, right? Things get wound up in derelict fishing gear or inappropriately placed out of season fishing gear. These things are designed to catch and kill things, right? Long lines, crab pots, it's all designed to catch things, and so in some cases actually do the dispatching for you. In large animals such as whales, this big, heavy anchor on your back, on your tail, on your fin, across your mouth can slow you down. It can make you worse at finding food. You can't keep up with your pod. You starve to death or get an infection, among other things, right? This is what happened to this killer whale. When we see things like this happen, it's important to understand more about the individual, right? In this case, this entangled whale, who is this? And this is important, especially for killer whales, because there's a lot of different kinds of killer whale. They're called ecotypes. So basically, different kind of population groups of killer whales all around the world specialize in different food items, behaviors, and stuff like that. They're so divergent that certain ecotypes have developed distinct dialects, as in they can't talk to other ecotypes. And this kind of split goes far back as tens of thousands of years, leading some scientists to think that they are separate species. And certain kinds of killer whale are endangered, such as the southern resident killer whales, the ones that occupy the Salish Sea, Puget Sound near Seattle. That's a very famous subgroup of killer whales that's endangered, given population depletion. While some ecotypes are easily definable and diagnosable, look at the Antarctic B2 killer whale on the left side, right? That looks quite different. It also lives somewhere quite different. But look at the killer whales on the right-hand side. Bigs or transient killer whales are the mammal-eating killer whales. These eat seals, sea lions, other whales, right? The resident killer whales are fish-eating specialists. They primarily subside on things like salmon, flatfish, things like that. Uh, while there are morphological differences across the ecotypes, if you looked at that from a boat, you could imagine how difficult it would be to actually suss out what type it is. The southern resident killer whales are very endangered. The bigs or the transient killer whales are not. So you could see how this can kind of feed into, well, we got to understand how this happened and who this happened to. So 2 weeks later, right, no one was actually able to get a tissue sample from this whale. The first report we got of this was from a fishing forum of all places. Wasn't actually directly given to us until quite a bit of time later. But 2 weeks after that, the whale shows right back up 2 hours south of Newport in a place called Bandon, really offshore of Bandon, Oregon. No one was able to identify the whale from external morphological characteristics. No one was able to get a tissue sample. No one was allowed to get a tissue sample. You don't have the proper permits as an everyday citizen, right? But thankfully, a Good Samaritan cut the entanglement line off of the animal. You can kind of see it here hanging off the skeleton still. So they cut that free, grabbed the pot and the line, and turned it into us. And that's where I come in. So I used DNA metabarcoding to first see what DNA was on this line, right? We had no tissue samples from this whale. It really stopped short with me to see what would happen from this story. Either it would remain a cold case and we wouldn't know who this whale was or where it came from, or we could figure this out. So using the short DNA metabarcoding fragments, I was able to get DNA from barnacles, from rotifers, and killer whale. Basically took a little bit of the green entanglement line, put it in like a DNA buffer, and let the DNA kind of soak off of it. And that's how I got those fragments. We did some more testing and got some more cetacean specific DNA in order to diagnose this to an ecotype. And this is really where the utility of metabarcoding can come in. This isn't really truly eDNA, right? It's not loose DNA fragments in the water. It's from a source. It's from the line that the whale rotted onto. But I want to think about this, right? Do you guys remember Jurassic Park? Do you remember Mr. DNA from Jurassic Park, the little cartoon guy who explained how they cloned the dinosaurs? If you take a DNA fragment and you read once a second for 8 hours a day, it would take you 2 years to read the entire DNA genome. It's actually 285 years is how long it would take to read a vertebrate genome, which is 3 billion base pairs long. Most of that 3 billion base pairs doesn't really matter for species diagnostics. We use short 200 to 500 base pair fragments, and that's where the magic happens. We're sort of min-maxing our ability to detect species at a high enough resolution to separate them out. But who is this whale? What was this whale? Stick with me. It's a lot of stuff and a lot of text on this page here. In the right-hand side, or left-hand side, my bad, this is a phylogenetic tree. The closer two points are together on these nodes, the more related these are. And each point, each label you have here is a different DNA sequence belonging to a different type of killer whale. So in this kind of gray complex in the top left-hand side, we have a cluster that represents the resident killer whales. So the fish-eating killer whales are more closely related to each other. There's also a bottom node there that says off. That's the offshore killer whales. These are shark specialists. We don't know too much about them yet, but we know that they have affinities to eating sharks, rays, and other elasmobranchs and stuff like that. In the blue bottom right-hand corner of this phylogenetic tree, we see a complex made of the transient killer whales. And in that red box in the corner there is our killer whale. Our entangled killer whale nests firmly inside this transient complex. So we have evidence and enough evidence to corroborate that this was a mammal-eating killer whale that got wrapped up in a crab pot and died. This doesn't happen often, as I mentioned before. I didn't want to declare this in the paper, but I think it's actually the first crab pot entanglement of a killer whale ever recorded in stranding history. Now, the stranding network goes back some 40 years at this point, so we had to do some digging to really, refine that statement, but this doesn't happen often. And so this leads to more questions. Why? Will this happen again? What can we do to stop it from happening? And science never stops, right? You're going to see this a few more times over this talk that science never stops. I'm going to take a quick coffee break. I don't know if we want to break any questions that came through, Katie, but if not, I'll just keep going after taking a sip of some stuff, some coffee.
Yeah, sure, Charles. Thanks. So, a question from the audience. Why use the term killer whale or orca?
Ah, great question. So, killer whale, right, refers to Orchinus orca. As I mentioned before, there are so many different kinds of orca, right? Genus name is Orchinus, the species name is orca, right? If all those different branches become different species, they'd be Orchinus something else, Orchinus something else. So calling them all orca is a bit of a misnomer. And really, honestly, this is from my heart. They're awesome. They're killer whales. Come on, man. You know, there are very few titles as distinguished as a killer whale. And it's really preference at this point. When you say either name, people know what you're talking about. But if you're a scientist like me, Orchinus orca, killer whale, who really cares?
Thanks, Charles.
Yes. I think I'm ready to get going on the next part of this talk. If you have any more killer whale specific questions, we'll answer them later. But let's jump onto the opposite coast. So this was off the coast of Oregon. We're going to go all the way to the East Coast. I visited here 2 years ago, had a red-eye flight out of Portland, got into Boston, had to learn Boston roads and drive all the way up Cape Cod to research and help with research on the North Atlantic right whale. This is a critically endangered species of baleen whale, the Mystacete whales. There's only about 300 to 400 of them left, though this year I think we had a pretty good calving year. There's like, I think somewhere in the neighborhood of 17 new babies in this population, so let's hope that this is good for them. Now, these whales are critically endangered for a number of reasons. Back during industrial whaling, the period of like the Industrial Revolution and stuff like that, we used whale oil for just about everything. Lubricating machines, making candle wax, food additives, you name it. It took the development of synthetic stuff or other sources of stuff to replace whale oil. Right whales were the right whale to hunt. They're slow. If you see in the GIF in the middle, that whale is surfacing, right? It's such a large animal. These things are a sizable creature, but only so much of it pops over the top of the water at any given time. And so they're also roadkill in the ocean, unfortunately. Large vessels hit them, they get caught up in entanglement lines. Somewhere in the neighborhood of 70 to 80% of adult North Atlantic right whales have been entangled or struck at least once in their life. And with a population of only about 400 animals, that is most of them. We know most of them by like an identifier. So it's important that we use more methods to study them and understand them, especially non-invasive methods like eDNA, right? One of the biggest prowesses of eDNA is that we don't have to see the organism. We don't have to touch it, right? Whereas with more direct tissue sampling, you have to go in and take a little pinprick and get something out of an animal or use a whole animal in some cases to get DNA out of it. Or in this case, we don't have to do that. For this research, we partner with an organization called Center for Coastal Studies. They're based out of Provincetown, Massachusetts, and from this area has been born a lot of right whale research. And so we're very thankful to work with them. Every year, much like other baleen whales, North Atlantic right whales undergo an annual migration. How the baleen whale life cycle works is that you have your babies somewhere nice and warm and protected, and then you travel up somewhere nice and cold and productive. That's where a lot of the phytoplankton grows, that's where a lot of the prey grows, and that's where you feed. So you spend a portion of your year eating and then a portion of your year breeding. For the North Atlantic right whales, they hang out around, Florida, the Gulf of Mexico, having a nice warm time with their babies down there. And then they travel all the way up to New England and kind of eastern Canada to feed, and the kind of Gulf of Maine system here. Migration is a natural phenomenon. It happens across so many different clades, from birds to insects to whales to humans. It's a natural phenomenon and should not be thought of as anything besides just organisms trying to live a better life and do the things that they have to do to survive. Right here is Cape Cod, Massachusetts, kind of I don't know, the top, not top 60%, but that 60% mark on the northbound migration route. Off the coast of Massachusetts, or actually along Massachusetts, is this kind of swoop of land that kind of creates a bay, Cape Cod Bay. And here is a principal foraging location for right whales. It's a great place to site them. They come here frequently, and a lot of research has been born from this location. And CCS is actually headquartered. You see that HC in the top right-ish of this map? That kind of like little divot of land is where they're based out of. And so they get to monitor this entire space every year for right whales to come in. It's federally protected. It's a great learning laboratory. And this is one such experiment that we did in 2018 and 2019, where we visited each of these labeled 16 stations. These are fixed points in Cape Cod that represent, different parts of the bay, right? You have parts that are more northern, more southern, more inner, more outer, right? You get a more comprehensive snapshot of Cape Cod, right? And taking eDNA samples from this can help us better understand the dynamics of this system, right? We are taking a seasonal picture of Cape Cod Bay using eDNA to see who lives there, when, and why, and how. And so obviously as scientists, we want to know what the trends are. Obviously as scientists, we want to know what did we find. Now we're going to go over some more fancy science graphs, so stay with me. So right here, each point on this graph, you see January through June. I've projected January 2019 to June 2019, which is part of this experiment interval. Each dot on this map, on this plot here, is a different station, representing different counts of different kinds of things found in Cape Cod Bay. The green is algae and phytoplankton. The blue is just kind of general animal species. The purple are prey animals for right whales. And the yellow are kind of other things like protists and miscellaneous things that I don't really understand too well. Much like how we observe in terrestrial systems, right? When winter comes and things happen, a lot of plant life tends to shut down or die off. Algae and phytoplankton are the plants of the ocean. This is where primary productivity occurs, right? As the season comes on, you start to die off, and then when the sun comes back out and conditions are more favorable, you grow back. And this seems intuitive, right? But it's just kind of interesting that you can observe this without needing to do anything much more than collect seawater, which is what we did. 16 seawater samples every month for 8 months, and these are the things found in those samples. Next up, we see the prey complex. Right whales feed on three copepod species in Cape Cod Bay. Well, three copepod genera, really. We're going to go into some fancy science terms. It's technically three genera. Copepods are these small zooplankton shrimpy things. You may have heard that whales like to eat krill. Krill is only one kind of small shrimpy thing in the ocean. The big baleen mysticete whales rely on these, millions of these in a single bite in order to survive. And so when we see the trends of these animals go up, it's around March and they persist through May and they peak around June time. It just so happens I didn't have the chance to make this look pretty, but stay with me. That on the top left here, we have a plot of the aerial sightings of right whales during this same period. You're on a plane, you count how many right whales you see in the Cape Cod. And the rise of phytoplankton, the rise of copepods match with the rise of instances of North Atlantic right whales in Cape Cod Bay in this period. Isn't that phenomenal that you can get all these kind of links together through eDNA? Now, obviously, correlation does not mean causation, and a big part of my job is to try to quantify and rule out other confounding variables and things that could be happening to make this trend behave the way that we see it. Though, based on my literature reviews, based on the models that I will not go into at this time, this seems to jive all together as legitimate seasonal flux of prey availability, which is awesome to see through eDNA because, again, we don't have to go through collecting of animals or algae, anything like that. We're just looking for their DNA signatures. And I don't know if any of you have tried to identify a copepod or a phytoplankton under a microscope to a species level, but it's not fun. It's certainly not fun at all. With genetics, we could do this no time flat. Now, you might be asking, okay, great, where's all the right whale DNA in Cape Cod? If you're seeing their prey jump up, where's all the right whales? And that's actually a bit of a quirk that we're finding with eDNA. Is that in a system like this, especially with the kind of study design that we have, remember it's 16 fixed points. We're not going anywhere else. We're going to these places and dunking a bucket in and getting some water, right? Vertebrate animals really like to move. And there aren't very many of them compared to invertebrates, right? You can envision that for every right whale in Cape Cod, there are literal billions of phytoplankton around it, just surrounding it in the immediate vicinity. Now expand that to like a square kilometer, and it's like a needle in a haystack. So we're trying to better understand the kind of dynamics of eDNA with large animals and why they don't shed as much. It's looking like it's an abundance thing, right? But it's very hard to prove that because there's always exceptions to these. Very, very much science, like it's typical. You come across a breakthrough and you come across more problems. But this doesn't mean that we don't get vertebrate eDNA in Cape Cod. Actually, quite the contrary. One of the most exciting finds that I got from this data set is we got white shark in October 2019 next to home base, which is very interesting to see. We also do get cetacean eDNA. I don't know if you remember that kind of aerial survey plot with the right whale observations. It really starts to ramp up towards February and March. And in February 2019, we do see a good amount of right whale eDNA in Cape Cod. There is something about the transport of water too that we're trying to understand about eDNA, but again, there's a lot of smart people working to answer these problems. And we also get some other cetacean species such as the Atlantic white-sided dolphin. And I'd be remiss not to mention some non-cetacean marine mammals that we see here. We do see harbor seal. There are two species of seal in Cape Cod Bay. Well, two primary species. We have harbor seal and gray seal. We get harbor seals on this coast as well, but they might be a different subspecies because of the distance between East Coast and here. Though, TBD on that. I'm not a pinniped guy, but it is delightful to see our bag-shaped animals represented in this data set. And I hope that that whirlwind introduction of eDNA and how we use it and how we use metabarcoding and all these methods made sense, first off. And second off, I hope I was able to properly convey that all of this is very much a work in progress. I'm preparing to finish up my PhD and writing things up and stuff like that. But for everything that we answer, we get more decision trees, right? Killer whale entanglement, right? Great. We know what the ecotype is. But will we see more of this? Why did this happen? Right? For the Cape Cod right whale prey survey stuff. Are the trends we seeing actually corresponding to legitimate, ecological phenomena? And where's all the right whale DNA and can we properly address that? Right? And then you could see more and more trees. I just want to step on a soapbox a little bit and talk about this because I think a lot of people look to researchers and scientists for definitive answers to the questions we want to address. We have so many problems in our world that we're facing, and we want answers, right? But unfortunately, the solutions and how to get to those solutions are quite complicated and convoluted, right? We, as researchers, offer an exploration of available options, and we see how far we could push the boundaries until we need to make either new tools or revisit existing tools and refine and make them better. It's never as simple as we want it to be. And I just want to say that just because I acknowledge all the work that's been going into stuff like this and in public health and in other ecological facets. And now more than ever that we need people around us in our communities to understand the work we do and why it's important. I care about whales. You probably care about whales. I care about the marine life. You probably care about the marine life. And everything is interconnected. Everything you saw here today has been applied to or will be applied to other ecosystems, other species, and it's only a matter of time before that happens if it hasn't already. So I want to thank my funders. A lot of good support has come through for these projects, including my other stuff that I didn't talk about today. I'd like to thank my lab mates, my co-authors, everyone who's helped kind of piece together these mysteries and help the scaffolding to address new questions. I'd like to thank Cape Perpetua Collaborative for being such wonderful hosts, and also my cat for not doing anything but being moral support during making this talk. And of course, I'd like to thank you for being here today and for being interested and curious about our natural world. And with that, I think I'm ready for our general questions.
Thanks so much, Charles. That was great and really informative. You're a great speaker. We do have a couple questions. So this first one is about eDNA sampling. So can you speak a little bit more about how eDNA sampling is done in real time in the environment where the water is moving? Is it different if you're in fast currents or slow currents or sheltered areas versus non-sheltered areas, shallow versus deep, you name it.
Well, I don't want to, undersell this, but there was a whole day in that 3-day eDNA symposium thing that was dedicated to this very question. It's like, how do the dynamics of eDNA sampling work? How does it affect your end result? But in the marine or any aquatic environment, what you do is you take a certain quantity of water. It's usually on the order of liters, 1 to 5 liters at a time. Some people go up to 10 liters, and you pass all that water through very fine, like mesh filters. These are little tiny filters. Oh, not little tiny. They're about this big, but the pores inside are on the order of microns. And to give you a perspective, that's how big certain organelles are in our cells. So these are made to capture those fine particulates of DNA and mitochondria and stuff like that. So you pass all that through your filter, zoop, all the purified, clean seawater comes out the other end, and you catch all the organic stuff on your filter. You extract that for DNA, and that's how eDNA sampling is conventionally done. There are other approaches too, very fascinating approaches that are being done and developed all the time. There's like real-time sequencing that's being developed. There's real-time like collection and PCR things that are happening right now. You could sequence on something the size of a Hershey's bar. That's how big some sequencers are these days. So yeah, there's a lot going on, but that's the general sort of theory behind it. And whether rushing currents and stationary stuff and how particulate heavy your system is, all of that does matter. And that's why you need to consider this as an integrative approach. I mentioned earlier that it was important that we talk to people like material scientists and engineers because they understand physics, right? And physical oceanography is a real thing, and everything affects each other and everything's connected, right? I will end with one funny anecdote. I was asked early in my PhD by a student from Alabama that, hey, I'm doing eDNA. Why do my filters keep clogging? Why can't I pass more than a liter of water? They were sampling in the Louisiana Bayou. And it's very particulate heavy, right? If you're sampling somewhere full of sediment and mud, that's going to clog up your filter. If you're sampling somewhere with lots of algae and stuff in the water, it's going to clog up that filter. If you're sampling at the bottom of the ocean where there's not a lot of stuff, you won't clog your filter. So there's that.
Great. Thanks for answering that. Another question about eDNA. Can eDNA be used to identify individuals or provide insights into how individuals are related?
100%. Yeah. So what I mentioned earlier is that we have short DNA fragments. On the longer end of this is around 5 to 600 base pairs, and that's not too long of a DNA fragment. Remember, the average vertebrate genome is 3 billion base pairs, but 500 is on the bigger side of metabarcoding. And generally, the longer your fragment size, the more diagnostic you can get. And there are different barcodes we could use for different clades of life and stuff like that. There are some barcodes that are very specific to one group of organisms. So for, in this case, let's consider cetaceans. We have some eDNA samples from Cape Cod from next to right whales, like in their immediate vicinity, that we're able to use for interpopulation level differences. We could see how different individuals are related, and we're increasingly getting closer to things like individual variation. But again, it's trying to min-max. You want a short enough fragment such that it lasts in the environment. Something I forgot to mention is that eDNA is degrading. The sun is degrading it, the temperature, all these biological and physical processes are occurring. The longer the fragment, the more likely it is to break apart. So that's why we target such small bits. So you want something that's small enough that it will last, but long enough that you can use for diagnostics. So we're getting there. Your mileage will vary on the kind of organism you're working with, but for elite cetaceans, we're getting to population level splitting. And hopefully soon we'll develop the probes and the technology to really consistently get individuals pulled out of that. And then you could do cool things like population estimates and stuff like that. That's what we want to move to.
Thanks, Charles. So now this question is about algae. So you mentioned how the Cape Cod algae and phytoplankton um, are seasonal. Was this the same type that we'd find in other areas, such as the Arctic? And why is the phytoplankton seasonal that you were talking about, but it can survive in such cold places like the Arctic?
Right. So different kinds of algae are adapted for different environments, right? So there are in very high latitudes, Arctic and Antarctic, very high productivity, actually. It's kind of surprising. These are kind of biological hotspots, despite being so cold. And it's a mix of things like nutrients, salinity, and all these other factors that play into how algae perform. So the kinds of algae and phytoplankton we get off Cape Cod are related to those in the Arctic because they're not too far away when you think about it. So they're similar genera. I'm not an algae expert, but they're similar genera, they're similar clades of algae. However, it's not the exact same species, though we do get something called Phaeostysis, like Antarctica something from Cape Cod, and it's not the Antarctic, but it's just the name of that algae. But also, understand this. I will not wish plant genomics on my worst enemy. Plants, algae, phytoplankton are so difficult to work with on a genetic basis because they could freely hybridize. They are multiploidy. They have all these weird characteristics that animals don't have. So we'll get there later, but this is to say that the algae we see in Cape Cod really boom bust with that seasonal cadence, and in the Arctic and Antarctic, those guys boom bust with their seasonal cadence, which is why climate change is such a problem, right? Things might not be able to catch up in time to face the new normals that we'll be facing. But who knows? Organisms are plastic and are able to survive in really bad situations. So I'm an optimist most days.
Depends on how much coffee you had, right?
Yeah, this is a 16 ounce right here, so we're only halfway done with it.
All right, Charles. So just one last question, a fun one to tie us all up here. What's your favorite marine animal and why?
My favorite marine animal, my fellow cetacean researchers are going to lampoon me for this, but it really has to be killer whales, right? You really just can't beat that caliber of animal, right? And despite there being so many, so it's like, okay, which one is really my favorite? They just have a myriad of behaviors. They're so complicated socially, and it's just awe-inspiring. I've seen a lot of whales in my day, right? I've seen blue whales, I've seen right whales, I've seen fin whales, humpbacks, bottlenose dolphins, North Atlantic, right? Yeah, all sorts of things. But the hairs on the back of my neck stand up whenever I see a killer whale. It's just that black dorsal fin, that distinct markings. I'm an artist, right? So when I look at nature, I'm looking for, striking things. And what better statement than to come fully dressed to every occasion in black and white? Come on. But I will let's choose something maybe a little uh, less loved here. My second favorite marine organism has to be my undergrad study species. This is called Tegula fudimbralis. This is a tiny purple snail that lives on the west coast USA. Um, they're humble. They're small. They don't do a whole lot. They eat algae. They like climb up rocks and stuff like that. They pop off rocks if you approach them and they tumble down. That's actually a predator avoidance behavior that they've evolved. And they're just nothing besides just the sentiment of it. Eric Crandall, we lost him a couple years ago and he was a good friend of mine. So whenever I see them, it really like puts me back in time to thinking about where I started. And I think that's important for all of us to think about where we've started, where we got our, first fix of the thing that we got passionate about, and where we're going to go next. So I I like to joke that I went from snails to whales, but really with eDNA, I don't have to choose.
Thanks so much, Charles. This has been lovely. That's a wonderful note to end on, and I'm sure your friend and former advisor would be proud to see what a long way you have come. So we wish you all the best of luck with your upcoming defense. I have no doubt that pretty soon we'll be calling you Dr. Nye. And, we look forward to staying in touch with you. Everyone else, thank you so much for coming and supporting our Young Wavemakers series. Once again, this is supported by small dollar donations. You can learn more on our website. The link is in the chat. We have really fun merchandise. There's a whale in our logo. And, we all hope that you follow us on Facebook, Instagram, and continue to stay in touch. Thank you, everybody.
Thanks,
Have a great
guys.
day.